So I am Pascal Cossard from the Pasteur Institute, where I am a professor and I am also a senior international research scholar from the Howard Hughes Medical Institute. This is the second part of my 3 talks, and I would like to discuss how Listeria monocytogenes has contributed to the new field of the new microbiology. So what do I mean by new microbiology? By new microbiology, I mean that there is a renaissance in fundamental microbiology because of the use of new approaches, and these new approaches are generating really new concepts. So the new approaches, the new tools, the new methods are really including genomics, transcriptomics, proteogenomics, imaging, live cell imaging, and single cell analysis, including and among others. The new concepts which are generated by these new approaches are, for example, new concepts in bacterial regulation, and most importantly, RNA-mediated regulation is really changing our view of how bacterial genes are expressed. Another new concept is the fact that bacteria are living in microbial assembly, either pure microbial assemblies or mixed microbial assemblies, not only with bacteria, but also with other microorganisms. The field of the study of bacterial communication is really exploding. Bacteria communicate via chemicals or via other signals, little peptides, and this is also something which is really increasingly important. Bacterial cell biology is also exploding, and imaging, which has been critical in cell biology of eukaryotic cells, is now also allowing us to have access to really details in gene expression, in how proteins are sorted in the bacterial cells, and bacteria are no longer considered as little bags, which are expressing proteins and other components. A new concept, which is really also increasing, is the concept of persistence, and this is particularly important in the field of bacterial pathogenesis, persistence and dormancy. Finally, the role of microbiota in health and disease is invading not only microbiology, but also immunology, neurobiology, developmental biology. So today, I would like just to talk about how Listeria has contributed to this new field of new microbiology. I remind what I said in the first part of my talk. Listeria monocytogenes is a bacterium which is present in the environment. It's motile at low temperature due to this flagella, but it's not motile at high temperature. So this is a bacterium which is ubiquitous and can easily contaminate food product. Contaminated food products lead to disease, and it's particularly important in immunocompromised individuals, old people, newborn babies, and also pregnant women. So the successive steps of Listeria infection are as follows. Bacteria arrive in the intestine via contaminated food product. Bacteria are able to cross the intestinal barrier. This is a unique property. Then the bacteria via the blood are able to reach the liver and the spleen. And then they disseminate to deeper organs such as the brain and the placenta. At the cellular level, the bacteria are invasive, which means that they are able to enter into cells which are non-phagocytic. So they enter into cells via 2 proteins which are called internalin and ENLB. Internalin is also called ENLA. Then the bacteria are trapped in the vacuole. They are able to escape from the vacuole thanks to the action of Listeriolysin O, a toxin which is called also LLO. Sometimes LLO is helped by 2 phospholipases, which are called PLCA and PLCB. Then the bacteria, free in the cytosol, not only divide, but they are able to move. And they move because they are able to recruit actin, polymerize this actin, and propel themselves in the cytosol. Not only they move, but they are able to spread from one cell to the other, and they generate, in that case, a two-membrane vacuole, which is lysed again by LLO and by the two phospholipases. So the bacteria disseminate from one cell to the other. And they do that by the factors that I have mentioned, and these factors are, interestingly, located on two main loci on the chromosome of the bacterium, and these genes are co-regulated by the protein PRFA. So PRFA is a protein which is similar to the cap protein of Escherichia coli. So the cap protein is the cyclic EMP receptor protein, and it's also called CRP. So PRFA is similar to CRP. PRFA co-activates all the genes which are on this slide. What is interesting is that at low temperature, when the bacterium is in the environment, all these virulence factors are not expressed. So we wondered what was the case, what was the situation for PRFA. And we could show that at low temperature, PRFA is also not present. But the message for PRFA is present at low temperature. And what we found is that the PRFA message at low temperature is having a very interesting structure that you see on this slide. So at low temperature, the message is forming this stem and loop structure, which prevents the ribosome to get access to the ribosome binding site, the Shine and Dalgarno sequence, SD on the slide. At higher temperature, this structure, these stem and loop structures open up, and then the ribosome have access to the Shine and Dalgarno sequence, and then the Shine and Dalgarno sequence allows the translation of the message. PRFA is made, and all the virulence factors are made in the same time. So this is a beautiful example of a new type of regulation of virulence factors at high temperature. In the last 2 years, there has been paper published which show that in the case of Yersinia, this is also the case, the virulence factors are expressed at higher temperature. This had been predicted a long time ago, but was really recently shown. In the case of Neisseria, there is also thermoregulation of virulence factors. So this is a really interesting property of Listeria. Virulence factors are regulated by a thermosensor. I would like now to come back to the post-genome studies. So we performed the sequence of the genome of Listeria to be able to have access to biodiversity, virulence, and global gene expression. So we very rapidly used high-density membrane. We could show that PRFA is not only expressing or regulating the gene that I mentioned before, but there are also all these genes that are on this slide, including the bisalt hydrolyse that I discussed in part 1. But we thought that we wanted to get access to the real transcriptome of Listeria, and we decided to use tiling arrays, not only to have access to the transcriptome of the genes that we had annotated during the annotation of the genome, but also to have access to the small RNAs because the field was exploding at the time. So we generated Listeria-specific tiling arrays, and we decided to look at the transcriptome of Listeria in many, many conditions. That is different temperature, in vitro, but also in vivo, that means in the intestine of germ-free mice, and also using different mutants. So I don't have time to tell you all the results, but I would like to tell you on this slide that this study has led to the identification of 50 non-coding RNAs. The number has now really increased. I will come back to that. But what was interesting in this study is that we could show that there were small RNAs which were not constitutively expressed. They were differentially expressed according to the conditions. And we had also some non-coding RNAs, some small RNAs, which were present in monocytogenes and were absent in Listeria innocua. For example, RLE38, which is present in monocytogenes, absent in innocua, highly expressed in the blood of infected animals, when deleted, is really affecting virulence. So this was one of the first small RNA which is affecting virulence. So we discovered a lot of things during this study, and in particular, we were interested in antisense mechanism. For example, and this was totally surprising, we discovered 3 long antisense RNA. These long antisense RNA are opposite to annotated open reading frame. We had also overlapping 3' UTRs. We also had overlapping 5' UTRs. And I must say that when we published these results, we were very stringent, and we knew that there would be other cases with structure which was not accessible when we annotated the genome a long time ago. So we decided that we had to go to another technique, which is RNA-seq. So we performed a whole genome transcriptomic. So that means we had access to, and we also used a sophisticated technique which allows to distinguish between transcriptional start site and processing site. So we could identify the transcriptional start site on one direction and the other direction, that means on one strand and the other strand of the DNA. We could also distinguish antisense promoter for antisense RNA and also transcriptional start site for small RNA, which are in between 2 protein coding genes. So this was a lot of data, and we wanted to give that to the scientific community. So we decided to build a browser. And the browser that we have generated is in fact a browser where not only we included the RNA-seq data, but we decided to put also the tiling array data and the tiling array data which were obtained by other groups. So altogether, all these data are available on the website which is indicated here. So that means if you go to this website, you can see how Listeria is transcribed in several conditions. And I'm just going to show you that you can go all along the genome and see how the genes are expressed. You see one promoter here, a promoter going in the other direction, and you can compare your gene with the gene of Listeria in different conditions. You see that this is a lot of data which are in this browser, and I would like to focus now on the antisense RNA. So I said already that we had been surprised in the tiling array data analysis that we had identified long antisense RNA. So in this RNA-seq study, we identified short antisense RNA and long antisense RNA. Concerning the long antisense RNA, there was one class which was really interesting. And this class is a class where you have a long antisense RNA, which is antisense for a gene, but which acts as a messenger for genes which are transcribed in the other direction. So in the example which is given on this slide, you see that this long antisense RNA is regulated by sigma B. So by using a sigma B mutant, we could show the effect of this long antisense RNA. And in a sigma B mutant, for example, we could show that the efflux pump was highly expressed and that the permease genes, which are in the other direction, were less expressed. So we came up with a new concept that we have named the excludon. So the excludon is a genetic locus where a long antisense RNA can act as an antisense and as a messenger RNA, regulating genes with opposite or mutually exclusive function. You realize that you can down-regulate one gene and up-regulate the genes which are in the opposite direction just with one long antisense RNA. And we have found that there are several examples in the Listeria genome and in other genomes. I think it's a new concept that you can only have access if you do RNA-seq. It's not by annotating a genome that you will have access to this type of data. So this is for the antisense RNA. Now I'm going to a new concept, which is riboswitches. So riboswitches, which have been discovered by other groups several years ago, can be classified in two classes, the translational riboswitches and the transcriptional riboswitches. So riboswitches are structures which are located in the 5' UTR, or in the 5' non-translatable region of messenger RNA. And these 5' regions can form different types of structure, depending on the binding of a ligand. So you see that if there is binding of a ligand, for example, S-adenosine methionine, SAM, you see that you have a structure which is changing, and in the case of the translational riboswitches, the ribosome binding site will be hidden and will prevent the expression of the downstream gene. In the case of the transcriptional riboswitches, if there is binding of the ligand, again, S-adenosine methionine, to the 5' UTR of the message, there is the formation of what is called a terminator, and then there is the generation of a sole transcript that you see here, which prevent the expression of the downstream gene. So riboswitches are generally regulating metabolic genes which are involved in the, for example, the biosynthesis of amino acids, but they are also involved in many other cases. So riboswitches can bind amino acids, they can bind tRNA, they can bind metals, they can bind different types of structure. I will come back to that in 5 minutes. So in the case of gram-positive bacteria, like Listeria, it's essentially transcriptional riboswitches which are in action. We found several unusual riboswitches when we were doing our tiling array analysis. So you remember, we were doing the tiling array analysis. That means we extracted RNA in different conditions. Then we were putting the RNA on the tiling arrays, and we were analyzing where we had transcription. And it was really amazing that we found several cases where we had riboswitches, downstream genes, and upstream of not upstream of open reading frames. So it was something that was a bit intriguing for us. But in the case of the B12, vitamin B12 riboswitch that I am showing you in the slides, we had another piece of data which led to a very interesting story. So as you can see here, the riboswitch, which is in green, is highly expressed in these conditions. And the gene which is downstream is POCAR, which is located in the other direction. But there is another small RNA, which is called RLEH, which has absolutely no transcriptional start site. So this was a really intriguing region. Not only it was intriguing, but it was interesting. Why? Because POCAR, the gene of Listeria, which is similar to the POCAR genes of Salmonella, is predicted to be a pleiotropic regulator. Indeed, in Salmonella, POCAR is involved in the co-regulation of genes which are involved in the utilization of propanediol in the intestine. And it has been very well demonstrated in the case of Salmonella that these genes are conferring a serious advantage to Salmonella compared to other bacteria when they are in the intestine. So we were wondering what was the situation in Listeria, and it was a postdoc in the lab, Jeff Mellin, who made this hypothesis that maybe this RLE39 was a riboswitch for a long antisense RNA, which would be antisense to POCAR. And to make a long story short, this is really what we found. This riboswitch is regulating an antisense RNA. So the model that we had in mind and that we have been able to prove is as follows. In the presence of propanediol and in the absence of B12, the long antisense is made. But as soon as B12 is present, B12 binds to the riboswitch leads to the expression of a short transcript, and the short transcript is not able to act as an antisense for POCAR. So we have a lot of POCAR, we have a lot of the PDU gene expression, and we end up to have a bacterium which is able to use propanediol if propanediol is present. So if I summarize the situation, the unusual B12 riboswitch in that region is regulating the expression of an antisense for POC-R, which is the pleiotropic regulator of the propanediol utilization genes. What I did not say is that all these genes are encoding for protein which require B12 for their activity. So you see that you have B12, which is regulating the expression of the antisense, and there is only expression of the propanediol utilization genes if B12 is present. So I think it's a wonderful example of the ending of a long story where we have done the annotation of the genome, we did the transcriptome by the tiling array, we did the RNA-seq, and now we are showing that the riboswitch is really regulating an antisense RNA, and this is in fact the second example of a riboswitch which is regulating an antisense RNA. So propanediol, where comes the propanediol? Propanediol is the product of the catabolism, is the product of, propanediol is made by commensal in the intestine, and it's a byproduct of different sugar like fructose and rhamnose. So really, propanediol is made by the commensals in the intestine. And surprisingly, it was the time where we were interested in the intestinal phase of the infection and the role of microbiota in the intestinal phase of infection. And what we had in mind was to decipher what was the role of the commensals on the expression of genes in the mouse intestine and on the expression of genes in the Listeria monocytogenes. So what we have done was a very, very reductionist approach, which is that we decided that we would take germ-free mice, axenic mice. We would infect them with Listeria monocytogenes in one case, or we would take the germ-free mice, put lactobacilli, and then infect with Listeria monocytogenes. And we wanted to study both the mouse transcriptome and the Listeria monocytogenes transcriptome. So we found that the lactobacilli are really affecting the mouse response. And we found that, in fact, the lactobacilli are protecting the mice against the Listeria infection. As you can see, there is an effect of the lactobacilli on the efficiency of the infection. But what I would like really to highlight here is that, in fact, there is an impact of the lactobacilli on the Listeria gene expression. And among the genes which are really affected by the presence of bacteria in the intestine, we found that the propanediol catabolism genes, the PDU genes, were among the most highly regulated by the presence of the lactobacilli. So this is really to show you that these utilization genes are critical for the survival, for the persistence, for the infection by Listeria monocytogenes. And what is really interesting is that among the highly regulated genes in the intestine, which are really regulated by the fact that if you pre-incubate with lactobacilli, you reshape the transcriptome, you also end up with the ethanolamine utilization gene, which are also highly regulated. And if I tell you that, it's because we are, at present, working on these ethanol utilization genes, and we are also demonstrating that there is a riboswitch regulated by vitamin B12, which is regulating the ethanolamine utilization genes. In fact, there are many, many riboswitches which we predict will be able to regulate non-coding RNA, antisense RNA. And we have performed bioinformatics studies, and we could show, and this is not highly visible on this slide, we could show that there are many isolated riboswitches which are not present upstream of open reading frame, but upstream of other of reading frame which are oriented in a different orientation. We have also found that there are many riboswitches which are very far from the downstream open reading frame, and we really predict that there will be other antisense non-coding RNA which will be regulated by riboswitches in many, many bacteria. So with that, I would like to conclude that by studying how Listeria is behaving, either in the mammalian cells or in the total organism, we have discovered a lot of new type of bacterial regulation. I would like to remind you that I mentioned the thermoregulation by an RNA thermosensor, which is regulating PRFA, the protein which is co-activating all the virulent genes. I've told you about the notion of excludon. And the excludon is this genetic locus where you have a long antisense RNA, which is acting as an antisense for a gene, and it's acting as a messenger RNA for genes which are located in opposite orientation to the gene which is regulated by the antisense. So in that case, the antisense is really acting as a messenger RNA. So I didn't have time to tell you about microbial assembly, bacterial communication, but I have shown you that one bacterium, the lactobacilli, are able to reshape the transcriptional program of Listeria monocytogenes inside the mouse intestine. And I showed you that the propanediol utilization genes or the ethanolamine genes are highly affected by the presence of the lactobacilli in the intestine. So with that, I would like to say that since 1986 that we work on Listeria, I think the field of microbiology has really changed, and Listeria has contributed to that.